Hello and welcome to this Nature Research custom media webcast titled Characterizing Oligomerization Dynamics of a Ubiquitin Ligase Using Mass Photometry. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Refeyn. Molecular glues are small molecules that stabilize the complex formation between E3 ligases and therapeutic targets. They facilitate the ubiquitination and degradation of target proteins, which are often considered undruggable in traditional drug design. Molecular glues prepare the surface of E3 ligase for ideal docking of the target protein. But the exact mechanism of action requires more understanding for effective molecular glue designs. This webcast will demonstrate harnessing the power of mass photometry to analyze the oligomeric state of an E3 ligase at low nanomolar concentrations. With us in the studio, we have Xiaojing Huang, who is the Facility Manager for Molecular Surface Interaction at the University of New South Wales. She'll run us through the applications and capabilities of mass photometry, how to use it to characterize oligomerization behavior of protein complexes, and the regulatory mechanism of molecular glues for targeted protein degradation. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to our speaker.
Hi. Today, I'll be talking about a class of molecules called molecular glue. I'll talk about what they are and how do we study their effect using native mass spectrometry and mass photometry. Before I start talking about the science, I want to talk a little bit about where the science takes place. I'm here based in Australia. I would imagine that's on the other side of the world for most of the audience sitting here today. But here in Sydney, it is really that beautiful, and I'm happy to do my research here and be part of University of New South Wales. I would like to acknowledge my PhD supervisor at the beginning, Alex Donald. Without him, this project would not have been possible. Now let's talk about why we care about those molecular glues at all. Conventionally, therapeutic drugs are often designed to target proteins with a well-defined binding site. For example, a drug mimics the substrate of the enzyme, or a drug binds to the ATP binding pocket of the kinase. However, it has been estimated that around 80% of the proteins can be considered undruggable in this way, because they don't have a well-defined small molecule binding site. They have a relatively flat and large interaction surface that is designed to interact with other proteins or DNA molecules. And this interface is typically dominated by polar and charged residues. It's different from what you find in the enzyme binding pocket, which is why it's so difficult to design a small molecule modulator to disrupt this interaction. Some of those undruggable proteins are transcription factors, or protein involved in transcription regulation, which is commonly altered in fast replicating cells, such as cancer cells. Here we'll be talking about one example of those undruggable protein called RNA binding protein 39, or RBM39. It is involved in pre-mRNA splicing, and it's regulated in many types of cancer. Research also shows that loss or inhibition of RBM39 can lead to aberrant splicing and eventually tumor regression. There are many approaches for those undruggable protein, such as biological strategies or gene therapy. We're talking about silencing the expression of those undruggable proteins. As you can imagine, this can have many downstream effects and is really difficult, but it is doable. And sometimes, in fragment-based drug screening, if a fragment hit a new mode of protein action, including covalent fragments or fragments binding to an allosteric site, fragment-based drug discovery can help develop drugs modulating protein-protein interaction. And of course, computational approaches are usually integrated into the target protein drug ability screening these days. At the end, I want to emphasize on this another type of approach called target protein degradation. This is the approach that's more relevant to today's talk. It's an idea proposed around 20 years ago about selectively degrading one protein using already existing cell machinery. But how does it work? We need to understand a little bit more about this specific cell machinery. What we're talking about is ubiquitin proteasome system. So after protein are made in cell, they're not kept around forever. The ubiquitin proteasome system helps regulate the protein level by degrading the unwanted protein and recycle them into amino acid, so we can use those amino acids to build new proteins. And it all starts with this small regulatory protein called ubiquitin, shown on the left. First, a ubiquitin is activated by E1 enzyme, performing a thioester bond between the E1 and the ubiquitin. Then the ubiquitin is transferred from E1 to E2. Now here's only a handful of E2, and they are responsible for recognizing a subset of E3 ligases. And when we move to the E3 ligases, we have estimated over 600 different types of rare E3 ligase, and they are responsible for recognizing different target protein and send them for degradation. Once this whole complex of E2, E3, and target protein is formed, due to the close proximity of E2 and the target protein, ubiquitin can be easily transferred onto the target protein. And after multiple rounds of transfer of ubiquitin, the target protein will have a tail of ubiquitin chain, and that will signal it for degradation by 20S proteasome. Targeted protein degradation utilizes this mechanism to destroy a selected protein. Well, how? We need to look at this complex. For a specific E3 ligase, it will have a native target protein. Now, if we add a protein degrader and initiate the complex formation between this E3 ligase and our target protein, in this case RBM39, in theory we can degrade any protein of our choosing in cell. But what are those protein degraders? Well, there are two major categories that people are working on. You have the bivalent proteolysis targeting chimera, the PROTACs, or you have the small molecule looking drugs called molecular glue. PROTACs have two protein interacting molecules. One binds to E3 ligase, shown here in blue, and another one binds to the target protein, shown here in yellow. And those two molecules are joined together by a linker. On the target protein side, it is shown here as a small molecule, but it can also be a peptide or a nucleic acid, and that will help recognize those undruggable proteins involved in protein-protein or protein-DNA interaction. PROTACs has a modular design, and that means a researcher can swap out the target protein interacting molecule with another molecules, and in theory, you can keep the well-studied E3 ligase and use it to degrade any other target protein. The idea of PROTACs was first proposed in 2001, and it's now the most intensively studied protein degrader, with two candidates in clinical trial, phase 2 and phase 3 already. But PROTACs development is not without its challenge, mostly the size of it. The modular design of the PROTACs also contributed to over 500 Dalton in size, and that characterizes it as a beyond the rule of 5 drug, with difficulties in bioavailabilities and pharmacokinetic properties. On the other hand, we have molecular glue. It resembled the conventional small molecule drugs that we're familiar with, fundamentally different from protein fundamentally different from PROTACs, molecular glue only interact with E3 ligase, and it prepares a favorable docking surface for the target protein. One molecular glue, thalidomide, it was actually discovered way back in 1957 and was marked as a drug for treating morning sickness. It was only way after they discovered that babies born after had severe deformities, and they didn't understand the mechanism for the drugs back then. It's way later in 2010, thalidomide was shown to act as a molecular glue, and it was actually degrading multiple proteins that are crucial for cell rapid growth. Now, thalidomide is actually the only clinical proved protein degrader for treating advanced stage cancer, such as advanced multiple myeloma. This case of protein degrader can be hard to design, but it's also the class we're interested in studying. In this study, we're trying to develop a method for directly observe the ternary complexes of E3 glue and target protein. We're aiming to answer these two questions. First, do molecular glue enhance the already exist but weak protein-protein interaction? And second, how does the molecular glue regulate the E3 ligase? A little bit about the system that we're studying. The specific E3 ligase we're working on is called DCAF15. It's DDB1 and Cal4 associated factor 15. It belongs to a Cal4 ring type of E3 ligase, and it's relying on this Cal4 ring subunit to bridge the E3 ligase to E2, which is the protein that brings in the ubiquitin. The construct that we're working on is a substrate receptor adapter complex, including subunit DCAF15, which will recognize a substrate protein, subunit DDB1, which is the adapter between DCAF15 to the rest of the E3 ligase, and DDA1, which is a small subunit that stabilizes the substrate receptor adapter complex. The molecular glue will bind to the interface of DCAF15, and it will initiate the interaction between the target protein and DCAF15. The target protein here is RBM39. The glue we'll be working on are a set of sulfonamides, including E7820, which is a molecular glue that's in clinical trial phase 2. The other three molecular glue work on is indicilin, CQS, and tacicillin. The main method we're using is native mass spectrometry. Mass spec is a familiar term for many people, but when most people think about mass spec, they think about a harsh condition where they digest the protein and look at the sequence of the protein. I want to emphasize that native mass spec is different. We're analyzing protein under a soft ionization process where the complexes and non-covalent interactions are preserved. So first, a fine string of tiny droplets is formed, followed by a rapid desolvation stage where ions on the surface of those droplets that carry the excess charges are ejected from the droplet. At the end, what we end up with is a naked protein ion in the gas phase with limited charges on the surface, and it preserved its native-like structure. What we do specifically in our group is that we reduce the size of the emitter from what commonly used 2 to 3 micron to 200 to 500 nanometer. This allows smaller droplet to generate it at the beginning, and that means there's less droplet heating during the desolvation stage, and that translates to less non-specific adductions and better preserved native-like complexes. Use native mass spec, we can directly observe the protein-protein complex formation with or without molecular glue. And that will allow us getting more information out of the single experiments as well, because we can identify all the different binding events. Another orthogonal technique we're using today is mass photometry. It is based on the principle of interferometry scattering microscopy, When a molecule is absorbed on the glass slide, the scattered light is corresponded to the size of that molecule. Mass photometry allows analysis of a single molecule at a low nanomolar concentration in solution, as opposed to gas phase in the native mass spec. Now let's look at our DCAF15 receptor adapter complex in native mass spectrometry. So looking here is the spectra of DCAF15 at 5 micromolar concentration. At the beginning, we're expecting one set of peaks corresponding to that receptor adapter complex. Well, as you can see, there's definitely more than one set of peaks. What we see here is DDB1 subunit and DDB1 in complex with DDA1. The worry here is that we're breaking the complex apart somehow in the gas phase, but we do later on figure out that we have excess DDB1 from the purification process, and the fact that we did not see any DCAF15 subunits on its own is also tells us we're unlikely breaking the complex in the gas phase. Moving on to the higher MZ range, we do see our expected peaks corresponds to those receptor adapter complex, but that's not it. At even higher MZ range, we see peaks corresponds to dimers and even trimers of those adapter receptor complex. Now, another study from earlier this year reported the same DCAF15 adapter receptor dimers and trimers, but at the time, this was the first time ever anyone observed those dimers and trimers. I want to verify those and even quantify those oligomers using an orthogonal technique, and that's when we move to mass photometry. Here, what we're looking at is 20 nanomolar receptor adapter complex of DCAF15 in mass photometry, which is a much lower concentration than what we just saw in native mass spec. We can still see this excess DDB1 subunit present in solution. It further confirms that native mass spec that I just showed you earlier is representative of what's in solution. Now, we did see DDB1-DDA1 complex in native mass spec, but the mass photometry here cannot resolve if DDB1 subunit is this peak corresponds to DDB1 alone or DDB1 in complex with DDA1. Move to higher molecular weight region, we do see monomer of DCAF15 receptor adapter complex together with the dimers and the trimers. This is indicative that we're looking at a self-assembled oligomer because we're enclosed at physiological concentration, and that this is the high affinity of oligomer because we are observing them at such a low concentration. To further quantify this interaction, I dropped the DCAF15 receptor adapter complex concentration all the way down to 3.5 nanomolar. And what we see is a concentration-dependent effect of oligomerization. At 4.3 nanomolar, we can barely see any trimer present, and at 3.5 nanomolar, there's almost all monomer with a very small population remaining in dimer. Based on this, I calculate the apparent dissociation constant for DCAF15 receptor adapter dimers and trimers to be 7 nanomolar and 9 nanomolar. Now that I've shown you DCAF15 self-associate into dimers and trimers, of course we want to know next is how molecular glue plays into this. Specifically that we know the monomeric form of DCAF15-DDB1-DDA1 is likely the active form, since the known crystal structure of the DCAF15 RBM39 in presence of one of the molecular glue E7820 show that DCAF15 is in its monomeric form. I use mass photometry to study the effect of molecular glue on the DCAF15 oligomerization state. When we look at the DCAF15 on its own at 20 nanomolar, the monomer diamond trimer have a relatively similar abundance. But when I add equimolar of target protein RBM39, there's no effect on the oligomeric distribution. When we add equimolar of one of the molecular glue, E7020, we see decrease in trimer abundance and corresponded increase in the monomer abundance. Now, if we add molecular glue and target protein together to DCAF15-DDB1-DDA1, what we see is the same decrease in the trimer abundance, but now we can also see a little bit decrease in the dimer abundance. We believe this is because the formation of active ternary complex with the monomeric DCAF15, and that's further shifting the equilibrium towards the monomer. When we express this in terms of apparent KD, we can see that the apparent KD increased to 16 nanomolar for the dimer and 26 nanomolar for the trimer, with just 20 nanomolar of E7820 present. And if we add both the protein and the glue together with DCAF15, we see that the trimer apparent KD remains similar to that with only glue added, but the dimer apparent KD shifted to even higher than with just the glue added. So we can confidently conclude that it is the glue and not the target protein that is modulating the oligomeric state of DCAF15. However, up to this point, we have not directly observed the ternary complex. The resolution of mass photometry cannot confirm if the monomeric peak we see here is just the DCAF15 monomer or the active ternary complex. To understand more about the ternary complex, we're going to move back to native mass spectrometry. Here, what we're looking at is the mass spectra of a mixture of 5 micromolar DCAF15-DDA1-DDB1 and 5 micromolar of the target protein RBM39 with 2.5 micromolar of the molecular glue E7820. We still see this excess of units, but what's more important is we see a change in the oligomerization distribution towards the monomer DCAF15. So in this monomer region, we can confirm that there's one copy of DCAF15 receptor adapter complex, one copy of RBM39, and one copy of the molecular glue. And that's just like what the crystal structure was solved before. In the dimer region there, we can see there's 2 sets of peak. One corresponds to 2 copies of DCAF15 binds to 2 molecular glue, and that is shown in triangle. Or there is another set of peak corresponds to 2 DCAF15 binds to 2 molecular glue, binds to 1 copy of RBM39, and that is shown in a purple circle. In the trimer region, we do not see any interaction between DCAF15 and RBM39. Here we prove that we can directly observe those ternary complexes using native mass spectrometry. At the same time, we can detect multiple binding events towards a different oligomeric state. We believe that RBM39 interacts with DCAF15 primarily as a monomer. To prove this, we decrease the molecular glue concentration. Here, what you look at is native mass spectrum of the mixture of 5 micromolar DCAF15-DDB1-DDA1, 5 micromolar of the target protein RBM39, and only 0.1 micromolar of the molecular glue 7820. We can see in the spectra that at low concentration of molecular glue, more DCAF15 dimers and trimers are present. In this dimer region, 2 DCAF15 binds to 2 molecular glue, and no interaction of RBM39 was detected. In the trimer region, same, 3 DCAF15 binds to 3 molecular glue, and no interaction is detected. But in the monomer region, we can see the majority of the signal shown in magenta is the ternary complex containing 1 copy of DCAF15, 1 molecular glue, and 1 RBM39. And a very low signal showing in orange, and it's probably probably easier to see at lower charge state or higher MC range. And we see some free DCAF15 showing orange. Based on these observations, I quantified the molecular glue effectiveness based on the abundance of the ternary complex, where the ternary complex is expressed as a fraction of all species containing DCAF15. Because RBM39 is only 13 kilodalton, the addition of RBM39 to a 160 kilodalton DCAF15 adapter receptor complex is assumed to not change the ionization efficiency much in native mass spectrometry. We analyzed the ternary complex formed at 3 different molecular glue concentrations, 0.1 micromolar, 0.5 micromolar, and 2.5 micromolar, plus at 0 concentration. We can clearly see a concentration dependence of the ternary complex formation. In total, we analyzed 4 different errors of phonomer molecular glue, E7820, indicilum, CQS, and tacicilum, with E7820 being the most effective molecular glue, and the tacicilum being the least effective. I also included a negative control at the highest concentration tested. The negative control is also aerosulfonamide, but does not initiate any ternary complex formation. The effectiveness of molecular glue is also tested by a solution phase technique called alpha screen. It is a plate-based assay with principles similar to FRET. In this assay, RBM39 is biotonated and conjugated to a streptavidin-coated donor bead, and DCAF15 is conjugated to the acceptor beads via the histacon DCAF15 subunit. When the mixture is excited by a wavelength, if RBM39 and DCAF15 is in close proximity, or in other words, they are in a complex, a singlet oxygen will transfer from donor beads to the receptor beads, and that will give a measurable emission signal. Based on alpha screen and multiple different concentrations of molecular glue, the EC50 is calculated from a dose response curve. Here, E7820 is also the most effective molecular glue with the lowest EC50. We take the ternary complex abundance from native mass spectrometry at 2.5 micromolar molecular glue and plot it with EC50 measured from alpha screen. We can see a nice agreement between these two assays. E7820 has the highest abundance of ternary complex, over 90%. It also has the lowest EC50, less than 1 micromolar. Tacicilum has less than 50% ternary complex in native mass spectrometry, and it also have the highest EC50, around 6 micromolar. This shows that we can use native mass spectrometry to quantify the molecular glue effectiveness rather than just identify the attractions. Native mass spectrometry can also provide additional information, such as multiple binding events at the same time. Now, I want to show that we can use native mass spectrometry to quantify those molecular efficacies, and it's not just an isolated case with DCAF15. So here I present a different system with cereblon as a different E3 ligase, or denote here as CRBM, and Icarus or IKZF1, Icarus zinc finger 1, as the target protein, and 3 different immunomodulator drugs as the molecular glue. And 3 different molecular glue concentration tested here, 10 micromolar, 5 micromolar, and 2.5 micromolar, plus at 0 concentration. We can see that pomalidomide is the most effective molecular glue between cereblon and Icarus zinc finger 1. Now what if we switch the target protein for the same E3 ligase? Instead of Icarus zinc finger 1, we now have Icarus zinc finger 2 and GSPT1 as well. This is the ternary complex abundance at 10 micromolar of molecular glue for 3 different target protein and 3 different molecular glue. We can now identify the target protein preference of cereblon Icarus-link figure 1, it's the most preferred target protein, and Icarus-link figure 2 is the least favorable. And what's interesting is the difference between Icarus-link figure 1 and Icarus-link figure 2 only differ by one amino acid. This target protein preference of cerebellum is the same as what is reported in the literature as well. Additionally, the molecular glue efficacy measured by native mass spectrometry here also agree with the reported affinity between E3 ligase and the molecular glue in literature measured by isothermal calorimetry. So native mass spectrometry can be used as a tool for studying those ternary complexes and is not limited to one E3 system. Now we only have one remaining question unanswered. Do molecular glue enhance the already exist but weak protein-protein interaction? Can we detect this interaction? Well, for those of you who are paying attention to the figures before, they might be a bit of a spoiler already. As you can see that at 0 concentration of molecular glue, there are some quantifiable ternary complexes. Well, here what we're looking at is 5 micromolar DCAF15 receptor adapter complex with 5 micromolar RBM39 in the absence of molecular glue in native mass spectrometry. We can see that the oligomeric distribution is mostly dimers and trimers, and RBM39 is not interacting with those dimers and trimers at this concentration in the absence of molecular glue. When we zoom into those monomer region though, we can now see that there's a set of orange peaks, and that is the dominant signal. It's belonged to free DCAF15 receptor adapter complex. At the very low signal, the red signal, and if you look towards the right-hand side at lower charge state or high MC range, we can see that the red signal corresponds to DCAF15 interacting with one copy of RBM39. To confirm such interaction, I increased the RBM39 concentration from 5 micromolar to 20 micromolar. Now it is in 4-fold stoichiometry access to DCAF15. We can see a set of relatively high abundance showing magenta that corresponds to DCAF15 monomer binding to one copy of our RBM39, and there's still some free DCAF15 monomer remains, and that's showing in the orange peaks. Based on the relative abundance and assuming similar ionization efficiencies, I calculated the apparent KD between DCAF monomer and RBM39 to be somewhere between 31 to 34 micromolar. It is important to know that KD calculated for protein-protein interaction in native mass spec is less accurate, but it can still serve as a valuable parameter in comparing interaction strength. Now, of course, we have to test this in another E3 system as well. Between cerebellum and 3 different target proteins, we observed interaction for all 3 pairs in the absence of molecular glue, with KD range from 35 micromolar between cerebellum and Icarus-link figure 1 to 162 micromolar between cerebellum and GSPT1. And the presence of molecular glue enhances affinity for all those interactions. In conclusion, through this study, I discovered that DCAF15-DDB1-DDA1 construct self-assembled into dimers and trimers, with KDs of 7 nanomolar for the dimer and 9 nanomolar for the trimer. And it is the addition of the molecular glue rearranged the DCAF oligomeric state to favor the active monomeric form. I successfully used native mass spectrometry to quantify the molecular glue efficacy, and the native mass spec method correlates well with established solution phase assays such as alpha screen. Finally, I provide evidence for the weak interaction between E3 ligase and the target protein. the presence of weak interaction is always a hypothesis in this field, but because the large material required to detect such interaction, it has not been widely reported. Now we have a method to detect these reactions with minimum sample consumption. Going ahead, we'd like to look at those DCAF15 oligomers in a bit more detail via either simulation or structural characterizations, or investigating if such oligomers are functional in cells. Another important aspect is, now that we know there is a weak interaction between the E3 ligase and target protein before we add molecular glue, what can we do with it? Well, those weak interactions can be used to identify a potential E3 target pair. Since molecular glue only have affinity for E3 ligase, for a specific target protein, if we can identify a few potential E3 ligase to focus our entry on, we can develop molecular glue for the target protein easier. At the end, I'd like to acknowledge my team. Here's us having a picnic right across the iconic Harbour Bridge. I'd like to thank BMSF for access to mass spectrometers. I thank Structural Biology Facility for access to mass photometers, ARC for funding, Triana as our industry collaborator, Refeyn for inviting me today, and Molecular Surface Interaction here at UNSW, which I am now a part of. With that, thank you, and I'm open for questions.